So today we are going to talk about foot and mouth disease, especially of cattle, and concept and benefit of essential oil blend for sustainable production of cattle. Foot and mouth disease, FMD, is a serious and highly contagious animal disease that affects all cloven-hoofed animals, including cattle, sheep, goats, camelids, deer, and pigs. Cloven-hoofed animals are those with divided hooves. It doesn't affect horses and zebras. FMD is caused by an aptovirus of family Picornaviridae. There are 7 strains: A, O, C, SAT1, SAT2, SAT3, and Asia1 are endemic in different countries worldwide. Each strain requires a specific vaccine to provide immunity to a vaccinated animal, and that makes it too much complex. Signs of foot and mouth in cattle: slobbering and smacking lips, shivering, tender and sore feet, reduced milk yield in milk cattle, sores and blisters on feet, raised temperature. Foot and mouth disease, the virus responsible for FMD is aptovirus. On the left side, the virion crystal structure is shown, and down is the genome structure. Gene expression and replication. The virion RNA is infectious and serves as both the genome and viral messengers RNA. The polyprotein is initially processed by the viral proteases into various precursor and mature proteins to yield the structural proteins, replicase, VPG, and a number of proteins that modify the host cell, ultimately leading to cell lysis. Ribosomal skipping is used as the junction of the 2A and downstream sequence. Viral infection steps. Number 1, attachment of virus to host receptors mediates endocytosis of the virus into the host cell by clathrin-dependent endocytosis. Number 2, upon endosomal acidification, the capsid releases VP4 that opens a pore in the host endosomal membrane, and the viral genomic RNA penetrates into the host cell cytoplasm. Acidic pH dissociates the capsid into pentameric subunits. VPG is removed from the viral RNA, which is then translated into a processed polyprotein. Shut off of cellular cap-dependent translation through the cleavage of translation initiation factors by the viral protease. Replication occurs in viral factories made of membrane vesicles derived from the ER. A double-stranded RNA genome is synthesized from the genomic single-stranded RNA. The double-stranded RNA genome is transcribed, replicated, thereby providing viral messenger RNA on new single-stranded RNA genomes. New genomic RNA is believed to be packaged into pre-assembled procapsids. Number 8, cell lysis and virus release. Number 9, maturation of provirions by an unknown host protease. Natural gut. Natural gut containing essential oils possesses pronounced capability to stimulate the induction of humoral and cell-mediated immune response of lymphocytes to the antigens of pathogenic bacteria and alloantigen. It maintains the essential metabolic function of an animal's body and reduces the stress level. It means the overall growth and productivity of cattle, which results in better quality meat and milk. The selection of blend oils done based on immunomodulating properties and their compatibilities to each other. The targeted active ingredients were as followed: Seniol extracted from eucalyptus, isobonal acetate extracted from pinus, and linalool extracted from lavandula. Then we talk about some concepts of the blend, synergism of essential oil blend. So each essential oil have multiple terpenes and sesquiterpenes that can be biologically active. So at time, there will be multiple chemical reactions, multiple biological compounds work together to metabolize these compounds, produces multiple biologically active components to fight against pathogen such as immunostimulant or immunomodulator. It is inhibiting and interfering with the specific pathogen structures and proteins such as membrane permeability. It enhances absorption of polar compounds across biomembranes, that is, carrier of the active herbal blend to its target. It reduces toxicity of the active compound to the organism while keeping its activity against the pathogen. Concept 2, I'm trying to show with the illustration and the color and the colors. This is showing how cellular functions are targeted. Right, red color balls are anti-stress response and anti-free radicals. Blue ones are anti-inflammatory. Yellow ones are the antiviral. Antibacterial ones are in green, and red and rose color are cellular signaling and apoptosis. Now coming to the computerized model in silico modeling. What is it? Natural antimicrobials could be effective in reducing viral and bacterial pathogenicity via direct or indirect antivirulence activities. The molecular docking or in silico model is a widely used technique to study a small molecular protein natural component ingredient interaction with the viral protein. So it's a protein-to-protein interaction at the atomic level. After calculating the binding position between the ligand and the target, the main purpose is to study the plant protein target viral protein interaction at the atomic level. In silico modeling, this way, molecular docking method allows screening of entire libraries, allows a precise antiviral, antibacterial component or drug development, and lead optimization. The in silico method also allows us to predict or help us to estimate the level of protection of each protein molecule present in the plant against the viral protein. In the present study, small molecule proteins of 3 plant oils, eucalyptus oil, pine oil, and lavender oils, were studied against the targeted proteins of foot and mouth disease virus. A small protein molecule were downloaded from the PBD website and opened in the PyMol software. As a second step, the potential proteins of interest from the FMD virus were downloaded and copied to the PyMol software. By scrolling and processing, it was allowed to bind both the molecules. To have a clear and fair study, the extra structures and water molecules of the protein were deleted by using the command. Only the interested domain of protein were used. The auto-inducer molecule were removed and the molecule of interest were lagged in. The protein structure was saved as LASR protein, which served as the receptor. As a second step, the auto-dock tools by NBCR was used to perform the protein docking. First, the small molecule that is protein of plant oils were loaded and then scrolled and searched the lag inside of protein with the small molecules. The values of docking score obtained of center X, Y, and Z axis were stored. The files were saved in PBDQT format and called as the docking score. Docking score is a computational result that is specific for the particular program and energy function, and that in an ideal case allows us to predict binding free energy and binding affinity, or to at least rank different complexes according to those parameters. The score mimics the potential energy change when the protein and lesion come together. This means that a very negative score corresponds to a strong binding, and a less negative or even positive score corresponds to a weak or non-existing binding. score. The score is listed in the docking results table as score. In the present study, the small protein molecules of the oils, such as linalool, eucalyptol, benzyl acetate, anethol, and others, have shown strong docking interactions with the viral protein. It also shows there is a stronger affinity of the viral protein binding sites with the plant small molecule proteins. The obtained docking results predicts that the active components of blend oils present in natural gut liquid are able to bind strongly at the receptor sites of the viral proteins. This is a strong way to deactivate the virus. Foot and mouth disease virus targets for antiviral strategies, which is FMDV RNA polymerase in complex with a template primer RNA, the crystal structure of the virus, and the structure of FMDV leader protease. Molecular interaction of FMDV protease with substrate and inhibitor. So FMDV leader protease bound to ubiquitin-like protein. And the second one is the leader proteinase in complex with inhibitor. Then molecular interaction of FMDV RD, RP with potential inhibitors like ketochromin D and quinadolin B. Now here we are taking some reference molecules and showing that there is a strong docking score, because you can see all in minus. Here also all in minus, they are strong. Again, all in minus, strong. Now we take the essential oil blend or natural guard major compounds docking score, affinity, and interaction residues. The docking score is a computational result that is specific for a particular program and energy function, and that in an ideal case allows you to predict binding free energy and binding affinity to at least rank different complexes according to those parameters. The score mimics the potential energy change when the protein and ligand come together. This means that a very negative score corresponds to a strong binding, and a less negative or even positive score corresponds to a weak or non-existing binding score. The score is listed in the docking results table as score interactions. The protein-ligand complex is a reversible, non-covalent interaction between two biological or macromolecules. In non-covalent interactions, there is no sharing of electrons, like in covalent interactions or bonds. What we are seeing, the plant molecules are also having a strong bond with the negative docking score. Here it continues. Knowledge about the affinity to the lesion is important, since the bilobal SPBs exist in equilibrium between the open and closed state in solution, and the addition of substrate directs this equilibrium towards the latter. A ligand is a small molecule that can bind to proteins by weak interactions such as anionic bonds, hydrogen bonds, van der Waals interactions, and hydrophobic effects. In some cases, a ligand also serves as a signal triggering molecule. A ligand can be a substrate inhibitor, activator, or a neurotransmitter. Here we can see eucalyptol, linalool, and anethol, all are having negative docking score. On NG oil, major compounds interaction with foot and mouth disease virus protease. Here again, we are using the reference molecules which are having high docking score. Continue. Then come to essential oil, natural guard major compounds. This terpineol, diethylphthalate, benzyl benzoate all have negative or high docking scores. Continues, eucalyptol, linalool, and anethol all have high docking scores. Essential oil, natural guard major compounds interaction with foot and mouth disease virus RDRP. Here also we can see the percentage is higher in the selected molecules. It's high. This work we also presented in international seminar organized by Elsevier. Then come to some in vivo trials. Here in this trial, which was conducted in UGM University, showed that the cattle fed on natural guard performed better in getting a higher weight gain after the transportation stress. Second trial is dairy cattle, which showed that the dairy cattle perform better in higher milk yield production. Even we see the level of different elements are higher. Coming to the next presentation, which was to show that this natural oil blend component can work against virus in vitro medium. This was also presented in Elsevier organized online seminar. This one is showing that that natural oil blend is having antiviral characteristics, and it showed its efficacy against African swine fever virus. This trial was conducted in Vietnam National Agriculture University, where pigs were challenged by the African swine fever virus. The obtained results from real-time PCR and clinical symptoms cumulative mortality showed that natural guard effectively inhibited African swine fever infection. The incubated challenge group showed that natural guard could deactivate the African swine fever virus in direct contact. The RT-PCR data showed that natural guard prohibited the horizontal transmission of African swine fever virus. The immunoglobulin G of natural guard consumed pigs was higher than the control groups. The immunoglobulin M value of natural guard consumed pigs was lower than the positive control. The work was published in highly recognized peer-reviewed journals. Now, a recent trial was conducted on foot and mouth disease in Indonesia. The trial was carried out in a village that were severely affected by foot and mouth disease in East Java province. Cattle that were infected for longer time under medication were 10. Cattle that were infected with FMD, fresh case with severe symptoms, were 4. Control was used in cases of remaining cattle in the village and from the surrounding village. Treatment was carried out using natural guard and natural guard liquid products from FMD infected cattle. All the animals from medium group were fully recovered. It is shown in the absence of physical symptoms of FMD that were originally appeared. The body condition were getting better, shown by the increasing on the blood body score and weight gain. Farmers had shared their testimony and were happy with the effect of products. All the trials were tested by real-time PCR, and it has given very good efficacy. So our conclusion and recommendation: on the basis of above trials and observation results, we could conclude that essential oil blend formulation is able to enhance and maintain the immunity of cattle and other animals. Essential oil blend make animals stronger to perform better against the viral and bacterial pathogens without having any significant negative impact. We strongly recommend to apply essential oil blend in the cattle for the better survival rate and productivity. Our special thanks to the governor, then Bu India, and Dr. Iswayudi of East Java Province, CPP Management, and RIA Management. Thanks a lot.